The Helping Interdisciplinary Vocabulary Engineering system is used to manage the ontologies and reserved vocabularies for disciplines. In any collection, the meaning of terms used for descriptive metadata may drift over time. At the start of the term, students assign metadata attribute names and select words to use for the attribute values. By the end of the term, the purpose for the collection has evolved, and the terms that are used for attribute values have changed. Some of the changes are driven by evolution of the intent of the metadata attribute requiring different descriptive terms, but the interpretation of a descriptive term may also change. An approach to minimize this problem is to use reserved vocabularies for the metadata attribute values. The terms are arranged in an ontology specifying relationships with other terms. The Hive system is available at cci.drexel.edu, MRC Projects Hive. The project provides a system for defining standard ontologies for multiple disciplines. Each registered term is given a URI that defines the meaning. The Simple Knowledge Organization System SKOS encoding is used to define relationships between terms. The Hive system can be accessed to verify the validity of a URI for a term or to check whether a term has been included in an ontology. A rule for accessing the Hive system is available in Chapter 5.12 of the Policy Examples Workbook. This rule checks the validity of term URIs. The ontologies registered in Hive define both broader concepts and narrower concepts for each term. In the example shown here, the term astroparticle physics has an alternative label, particle astrophysics, and is linked to broader concepts such as interdisciplinary astronomy and to narrower concepts such as gamma rays, cosmological neutrinos, and gravitational waves. For each concept, a registered URI is provided that is associated with the concept label. The vocabulary for the set of concept is also given a name, in this case UAT. If you look at the narrower definitions for each label, there is a unique identifier specified within the UAT vocabulary. Hence, gamma rays has the unique label T635, gravitational waves has the unique identifier T689. Thus, every term has a unique identifier that can be related to other terms within the SKOS vocabulary. The ontology can be searched to discover if a term is registered. A dictionary of well-defined terms can simplify assignment of descriptive metadata. Terms that are used for description can be pulled from the vocabulary. This ensures the existence of a knowledge community that understands the vocabulary, will also understand the context for the items in your collection. There still are challenges. These include interpretation of the context by researchers from other disciplines and interpretation of the context by future researchers. The ontologies themselves will evolve over time. Take 7 minutes to complete exercise 60. Decide whether a preferred metadata schema exists for your research collaboration. Look at hive.cci.drexel.edu for examples of registered vocabularies. Which discipline vocabulary is closest? UNC has multiple projects that generate genomes. The project managers realized that a valuable resource could be created by depositing the genomes into a common collection. This would facilitate additional research, serve as an archive for their data, and provide a way to publish data. The requirements for the formation of the genomics data collection were that minimal impact would be made on the existing projects. While researchers were willing to share the genomes, they still had to complete their existing research grants and could not put significant effort into the common repository. An extensible approach would be followed. New projects should be able to participate with minimal effort. A distributed data environment would be supported. Each of the participating sites had stored their genomes at different storage locations using different types of storage systems. The approach that was followed was to use a policy-based data management system to register data from existing repositories into the common collection. A reference is shown here, renci.org, WP-content uploads, and then a PDF file. The processing steps applied to genomics data involve multiple repositories. The original data comes from sequencers and are saved in tape archives. The original data are then processed through an alignment pipeline with results again archived. Genome variants can be detected with information submitted to a variant database. The data may then be accessed for use in clinical decision and support planning. If a common repository is created, it will have to retrieve data from each of the archives, identify which stages of processing have been applied to the data, track access controls for proprietary data, and maintain links back to the original research project. The architecture used at UNC to implement the genomics data grid is shown here. A secure medical workspace supports processing of patient data. The only information that leaves the secure medical workspace has been appropriately anonymized. Information from sequencers are deposited into an iRODS data grid and written to disk for processing in parallel with a Hadoop computing system. Data are written to a secondary storage system for additional processing steps. The addition of access to data from another project is accomplished by installing DataGrid software as an application on the project's data server. This makes it possible for the project to continue to do their own analyses, but enable incorporation of data into the common collection. One advantage of the common collection is that it becomes possible to implement a unified view of the data. The files can be organized into collections that are independent of the actual physical storage location. Thus, data on disk storage at UNC, data on disk storage at RINC, and data on tape storage at RINC will appear to be in the same collection. This greatly simplifies access since any of the datasets can be retrieved using the DataGrid clients. The same naming conventions, collection arrangement, and access mechanisms are used across all of the storage systems. The UNC Genomics Data Grid is a prototype for clinical genomics projects. These have the same challenges in using genome data in clinical practice. Genomes have to be named, arranged, and accessed across multiple locations. The processing applied to each genome needs to be tracked. Access controls on protected data need to be honored. The system has to scale to 100 million genomes distributed throughout the healthcare system. A project which is looking at these issues is the North Carolina Genetic Evaluation by Next Generation Exome Sequencing. This project manages the same processing steps described earlier.